So I'll call this video Endocytosis the big picture. We've talked about the little picture, which is the nitty-gritty of the trafficking reaction of endocytosis. But now I want to deal with what is probably pretty obvious, and that is that endocytosis is quite a bit more complicated than the other trafficking pathways. And in fact, it is a branched pathway, and that is an indication of multiple functions associated with the endocytosis. So when we talk about COP2, we're talking about secretion mostly, and likewise exocytosis, although not always. But COP1 pathways are pretty much retrieval, but endocytosis is involved in a bunch of functions, and that's one of the reasons for its complexity. So this diagram is taken from a previous video in this original article, and it shows sort of the pathway nature of that. And one of the principal functions that we attribute endocytosis to, probably the most commonly thought of one, and that essentially is nutrition, in which things that are brought in from the inside go through a series of vesicular maturation and translocation processes, ending up in the lysosome, where things are reduced to their molecular nature and then released into the cytoplasm, essentially as food for the cell. So that process, yes, would be appropriately referred to as nutrition. But depending on what the thing that is brought in is, it could also be considered immunity. That is, this might be a virus or a bacterium. It might even be an intact eukaryotic cell that is brought in through this pathway. So we can go ahead and add immunity, shall we call it, as a function of the endocytic pathway. Another function is associated with this particular set of reactions, going from what's referred to as the early endosome to the recycling endosome, and then potentially right back to the plasma membrane or maybe to the trans-Golgi network. I would refer to that as upkeep, basically, or recycling. And the reason to call it that is because this process is an extremely rapid and common process, and it involves cargo of all sorts, including the lipid of the endocytic vesicle membrane itself, in which the contents of the plasma membrane are essentially replaced on an hourly or maybe daily at the longest basis, such that the entire lipid composition of a plasma membrane undergoes a recycling process, such that lipid is removed and then replaced on a continuous basis, and that keeps the plasma membrane very nice and shiny, apparently, because pretty much all eukaryotic cells undergo that as a very active process. And then finally, I think the last thing that we'll talk about is the involvement of endocytosis in signaling reactions. And the reason to put it last is not that it's important, it's just that signaling is the next big topic that we're going to talk about. Okay, so the way to break that down is, well, there's no logical way of doing it, but what I'll do is talk about the lysosomal pathway first, and that leads us into a side process that ends in the lysosome, but actually doesn't begin with endocytosis. That's autophagy. And then we'll go back to things that really do start with endocytosis, and that includes the so-called multivesicular bodies that I've alluded to earlier, but we'll talk about those specifically. And then exosomes, which can be thought of as a product of the MVBs and are very important in the signaling reaction. So as far as a breakdown of these categories, this figure of 1342 of your text does a good job of that. So here's pretty much what we talked about last time: the budding and coding and uncoding and fusion, ultimately, of the thing here that would be referred to as an endocytic vesicle, and it will either become or fuse with something that would be identified as the early endosome, and then recycling endosomes would pop off that and lead to the replenishing of the membrane, etc., etc. So this would be an example of something, the uptake of a small molecule nutrient or small molecule nutrients, solutes, that would be referred to as just straight pinocytosis. And that terminology is kind of old school, but it is useful because there's another type of import of small molecules using endocytosis that looks like this. So this diagram is a little bit confusing if you don't stare at it a little bit. But this is the plasma membrane, and this is the extracellular space. So don't get confused in thinking this is the plasma membrane. So what's happening is that the plasma membrane is fusing right here and engulfing this part of the extracellular space, and that becomes the lumen of this endocytic vesicle. So this reaction, which is referred to as macropinocytosis, is capable of bringing in substantially larger things than pinocytosis would be able to bring in. But the fate, roughly speaking, is going to be the same. They end up in the late endosome and then ultimately get gobbled and chopped to pieces and dumped with battery acid in the lysosome. So that immunity function would be indicated by this part of the diagram here. Phagocytosis is the engulfing of very, very large things, big viruses and bacteria, and in the case of immune cells, other eukaryotic cells. This could also be an apoptotic body when we start talking about apoptosis. And that process is generally, if it works correctly, ends up with the bacterium or virus ending up in the lysosome where it's destroyed, and then its raw materials, amino acids, and maybe a nucleotide here or there, some lipid, gets distributed to the rest of the cell. So that's both nutrition and immunity by my way of thinking. And then autophagy, because this process ends up in the lysosome, is related to this overall topic, and I'll talk about it specifically in its own section. So let's zoom in on this part of the lysosomal pathway and introduce figure 1347 in your text, which is shown here. So here is the endocytic vesicle that is doing whatever it was that got to this point. It's not so essential that we know at this point. That leads to fusion or maturation into something that's referred to as an early endosome. And all of these intermediates can be distinguished by the internal pH, as that earlier diagram showed, and that's associated with the prototype pumping by the V-type ATPase, as I mentioned earlier. This is what you would see in electron micrograph, and you can appreciate how difficult it is to tell what exactly you're looking at, because this is an early endosome, but it, for all intents and purposes, could be a recycling endosome based on appearance. So you have to be a pretty imaginative electron microscopist to know one from the other. But in terms of their function, they're quite a bit different. The early endosome is sort of a clearinghouse for the vesicles that come in and go out, and the cargo, obviously, that does the same thing. So that there is some, within minutes, maybe this might be a 5-minute process that goes from the early endosome back to the plasma membrane, and there are some pretty good reasons for doing that I might or might not talk about, other than just lipid renewal. There's also a recycling pathway that goes to a recycling endosome, and that endosome either can fuse with the plasma membrane directly or become or butt off cargo vesicles that go back there and fuse. So I want you to pay attention to these things. I'll talk about those specifically, but those are interluminal or luminal vesicles that are the product of a very unusual interaction between the endocytic vesicle as it arrives and the fusion process, which includes an additional ingredient that puts it where it is. More about that later. The other thing to remember is that there is a microtubule association. Even though these things are very big, they're organelle-sized things, they are dragged along by motor proteins using the microtubules as the motor for that translocation. And then finally, in going from the late endosome, a very differentiated form of endosome, to the lysosome, it's really kind of a gradient, a gray area, which is one and which is the other. They're very similar But this diagram indicates that the interior luminal vesicle structures tend to be pretty elaborate by the time you get to the endolysosome. And at least in the early stages of the formation of a lysosome, which we'll talk about specifically in a minute, it's very difficult to distinguish the lysosome from a late endosome or an endolysosome, which is essentially just a lysosome fused to the late endosome. But eventually, all of these things are going to go away because they're going to be destroyed by all of these things that are present in the lumen of the lysosome and not in the late endosome, at least not in particularly active form. So very low pH, a bunch of enzymes that degrade things, so hydrolases, proteases, nucleases, glycosidases, lipases, that are activated by the low pH and by becoming activated, destroy DNA and RNA, proteins, the carbohydrate, oligosaccharides that some of those proteins contain, membranes, and all of the additional components that would be dumped into the lysosome. So when we say that nutrients come out, it's a result of a very, very active hydrolytic and oxygen radical and low pH associated degradative process that occurs in the lysosome. So these things are membrane enclosed, so those membranes are destroyed, and you may wonder, well, why isn't the lysosome membrane destroyed? And that's a very good question that no one really can answer very well. There is sort of a cytoskeleton associated with the interior of the lysosome that offers some of the protection, but much of that is kind of a mystery. And probably some of it does get destroyed because lysosomes tend to come and go very actively inside the cell.